Time to introduce, now we're going to actually, you're not going to hear from me anymore for a while. I'd like to introduce our first speaker, and actually we don't have that many speakers. I said we have mainly panels and breakout sessions. But David Rim is going to really kick things off for this meeting, and it's my pleasure to introduce him. David is an MD PhD, board certified pathologist. He also runs a research lab as professor of pathology and medical oncology at Yale University School of Medicine. David is also the director of the Yale Pathology Tissue Services and the Yale Tissue Microarray Facility, and serves as the director of translational pathology. His own lab group focuses on quantitative pathology using aqua technology. I don't actually know what that is, David, but maybe you'll explain it. But he invented it, or his lab invented it, and he particularly looks therapeutically at both therapy and recurrence of metastases in lung and breast cancers. So David is a real doctor. Not yet, Dave, not yet. The technology has been used in a series of efforts related to biospecimen science as well. I think that's of interest and importance. He also serves on the Molecular Oncology Committee at the College of CAP, the College of American Pathologists. He's an author of over 300 peer-reviewed papers, and he has 8 patents, and he serves on a lot of advisory boards for biopharma and diagnostic companies. David has been a consistent voice calling out for standards for research antibodies, and in particular, he and his lab published a widely cited paper in biotechnology on the topic a few years ago. David McAdams, I believe, referred to it last night. And when GBSI, when we started engaging on the topic of antibodies and antibody validation, I reached out to David. I didn't know David. I just knew his paper. And I think that in our first phone conversation, he really didn't know I don't think he'd ever heard of GBSI, and I'm not sure he'd ever heard of me. And I think he was very cautious, as he generally is, and didn't really know what to make of us. And I convinced him that we were absolutely serious and dedicated to addressing this issue and ultimately defining antibody validation. And I have to say he's been a tremendously invaluable member of our steering committee. And David, look where we are today. So thank you, and it's a pleasure to introduce you this morning.
So thanks, Len, for inviting me, and thanks especially for organizing this conference. I'm honored to be in front of you all. In some ways, I think you all, as a group, represent the solution to this problem. And I really, I look forward to working with everybody, hearing everybody's opinion, and trying to think forward about the problem, and not our own problems, but the global problem, so we can sort of move forward with science. So Len and you all, thank you for the time. I'm going to try to go briefly through a few things. First, even though this is not continuing medical education, I still need to show my disclosures. As Len pointed out, I do a fair bit of advisory board and consulting and funding from industry. However, I would point out that what I do in an academic lab can't affect patient care unless I have a partner, and these are my partners. So what I want to talk about today is start from the beginning. I'm sure many have heard about this. I guess my lab was the poster child for this problem, in that we had so let me tell you the story. Let me start by telling the story that really in some ways started it all, and then we'll move more quickly through some of the other issues. So I had a graduate student who and a desire to be able to predict which melanoma patients have metastases to their sentinel nodes and which do not, so thereby preventing the need to have a sentinel node biopsy. And the story is this, that if you have a melanoma somewhere on your body and you have a metastasis to your sentinel nodes, you have a very high likelihood of recurrence, and you will be treated differently and managed differently than if you do not have a sentinel node positive. However, of those patients that are sentinel node positive, or sentinel node negative, 2 or 3 are going to recur anyway. So those who we wanted to find. So the way to do this is we looked at large numbers of melanomas, training sets, validation sets, and we used these antibodies to determine which proteins, and we looked at about 400 different proteins to determine which proteins, when combined, could generate a signature model to predict response to melanoma, and we were able to do that. In 2009, we published a paper in the Journal of Clinical Oncology showing a 5 antibody-based quantitative measurement. That's what AQUA is. It's a way of quantitating measuring. It stands for Automated Quantitative Analysis, but subsequently trademarked by Historics. But the term is generically quantitative immunofluorescence. And so we used the quantitative immunofluorescence to accurately measure the number of, or the amount of protein of these 5 proteins, and then came up with an algorithm and a training set, and then validated and a second set, and then began the process of commercialization. And the first thing we did was go back to the antibodies that we bought originally and show that on tissue microarrays that they were the same as when we did this experiments 3 years ago. And 2 out of the 5 had no relationship to their previous data. And this is quantitative measurement. I showed, this is, for those who are not aware, this is a tissue microarray, H&E, and this is the quantitative fluorescence tools that we use to do that. And we showed that basically there was no regression. So this was really frustrating because at this point we'd already spent over $1 million on this process through R01 grants. And I had a second R01 grant, the highest rating I ever got, a first percentile, to do to the next step. And we were on it, and we were stymied right in the beginning. We began testing other antibodies. We began a long, sad process that ended in that project ending in my lab without anything to show for it, other than a couple papers. And this might have been more important than the papers that would have come out of it, because we have a chance, I think, to fix the problem. So this is not, were they bad antibodies? No, the antibodies weren't bad. They were bad management of antibodies, and they were bad management. It was a bad system. So I don't really think we have that problem. It was a system of irreproducibility. And Glenn Begley and Lee Ellis pointed that out to us in a big way, that we have a major problem in science right now, and Len talked about it, so I won't go over it again. But what causes that irreproducibility, or that lack of reproducibility? They had this survey in the Science, and I thought it was interesting that only 1 in 4 are able to publish results. And the key reproducibility issue, number 2, 36% antibodies. So we can fix a major part of irreproducibility may be due to this reagent that we're all so fond of and use so heavily. So this is a sort of a summary of some of the press that this has gotten more recently, some of the calls for antibody validation and why we're here. And this is from Matthias Uhlen's group, where they looked at the percent sex rate in external antibodies and actual validation, and it ranged from as high as 100% to as low as 10 or 15%. So really a lot of the antibodies out there, and this is my experience in our lab too, on average when we're going to work up a new antibody for some new biomarker usage, especially predictive biomarkers, my lab is more interested now in predicting response to specific therapies, we on average go through 3 or 4 different antibodies that do not validate. But if it's a unique biomarker, if it's a unique protein, it may not be available from some companies, and it may be available from others, and so we can't count on a single vendor, and so as a result of that, we struggle with validation, and this is an example of that. And I thought this was nice, sort of as a place to start. Matthias put together this sort of timeline of all of the papers and events, and we kind of look like we're in the middle here, which is good. I think this is a problem that's been a long-standing problem, and that we have a chance here to address and a chance to change the direction that we've seen in the literature. So how we got involved is after that mishap, a young lady in my lab, Jen Bordeaux, was having trouble with her thesis project, which was on MET, the CMET gene, or the receptor for this scatter factor or hepatocyte growth factor. Turns out the antibodies for that are a mess, and she was suffering the same problem, but it may be worse than the antibodies. It may actually have to do with antigen retrieval and folding of the antibody and lots of other problems. And in her frustration, sort of abandoned that problem and began with me working on what is now the paper that kind of got the ball rolling in many ways for our lab, which was to try to come up with an algorithm that this was our first pass on, was too complicated and too obscure to figure out, but at least this is what we did in our lab, so we shared it. And my highest cited paper is cited about 1,500 times. This article has been downloaded 35,000 times. So I think that there's interest. This tells me that this is a problem that people feel, and we're not the only ones that are suffering. People need to figure out how to solve this problem. So what I want to do in the last 10 or 15 minutes, since we want this to be interactive and not lectures of stuff you've already heard, is I'm going to tell you some stuff that some of you may not have heard and some of you have already heard. It's what to do, the science behind antibody validation. That is scientifically rigorous and reproducible data for quantitative analysis, for sensitivity, for specificity, and for reproducibility. And then I'll spend maybe 3 to 5 minutes talking about what we've done so far with the International Working Group and where we hope to go. So here's 3 examples of problems that we faced in our lab. One of my graduate students back in 2007 were interested in EGFR, and I'll start with that one. And she tested 5 different antibodies. Here's the antibodies here, and the suppliers are here. And we were thinking, okay, we're gonna get this figured out because we're gonna test more than one antibody on the same on the same exact patients, on serial sections of tissue. And this is what we got. We looked at 600 breast cancers, serial sections, and these are the pairs of antibodies quantitatively measured. One of them actually regresses. So 31G7, which was a polyclonal, and PDX, which was a monoclonal, showed some regression. All the rest didn't. Now, some of that might be because some were extracellular domain and some were cytoplasmic domain. So it might actually be biology. And in fact, that's true in MET and HER2 and in HER1. They go to different places because that's real biology. The molecules cleave and it changes. But some of it is just a problem of epitopes that are immediately adjacent that must cross-react with something else. And so this was actually published in this analysis where we looked at a bunch of other antibodies, including HER3, and then showed an EGFR as an example of how this does validate. Here's an example of EGFR. Here's 5 different antibodies. This is what it should look like. If there's not a lot of processing and you're quantitatively measuring antibodies, your regression value should be in the 0.85 to 0.95 range. They're serial sections. They're not identical pieces of tissue. That's why it's not a 0.99. And there's other variances and noise in the measurement systems. But this is our goal, to be able to use measurement systems to get this kind of reproducibility, especially when antibodies have adjacent sites. And we'll talk about that later because that's a potential validation method, is to use adjacent site antibodies. So what happens, though, in the real literature is something like this. So this is an example of a failure that led to a whole literature that's now in question, and that is the antibody MC20. So we took and if you look at it on a Western blot, it doesn't look bad. Unless you overexpose it a little bit, then it looks pretty bad. Here's another monoclonal antibody the same way, and the monoclonal shows progressively increased expression in an induced series along with this, mixed in with some cell lines. But the polyclonal is kind of all over the map. And then here's an induction series of ER in a closed system, or a recombinant system. So you can see increasing amounts of ER, and here's MC20. So the results of this is that there's a whole literature describing the cytoplasmic localization of estrogen receptor. Some of that literature may be correct. Some of it is almost certainly false because it depended on this antibody which binds to something else. This is more relevant and more recent. This was a paper on PD-L1, and this is unfortunately a major infraction in the pathology community. As pathologists, and I am one, many of my peers don't have their own lab. So they do work by buying an antibody and reading what's on the label, and then getting the tech in the main lab, in the histology lab, to do some work on the side for them. Just do 50 or 100 of these, and let's see how they come out. And that may or may not have been what happened with this group, but here they find an antibody against PD-L1, which is a known membrane protein. Okay, it could be in the Golgi, it could be in the estrogen receptor, but it's never been described in the nucleus. And so a very simple validation rule was violated, and they even showed it in their paper in Figure 1, a nuclear localization of PD-L1. So these are the kinds of things that we have to present, and those are and so how are we going to get there? Those are sort of some classic problems in the field. And so this, I actually wanted to show a picture of Jen. This is Jen Bordeaux when she was in my lab, and this was and she was actually the author of this flowchart. With obviously input from the rest of the authors. But what we can do is kind of walk through this flow chart in a simplified manner. So what are we really doing in that flow chart? Sensitivity, specificity, Specificity on the slide, because this is an IHC antibody validation, and I note this, if I say one thing that you all remember today, it should be this. All validation methods must be context specific. What works to validate on one technology may not work on another technology. And so as we come to this, we're going to have to remember to keep things context specific. So this is a context for immunohistochemistry, and it turns out quantitative immunofluorescence and immunohistochemistry have very similar behaviors and can be validated in the same way. And so that's specificity on the slide. This wouldn't necessarily be added in a context of flow cytometry or mass spec. And then finally, reproducibility, and this one's probably more important than anything else we do, because if this fails, it means that one of these will also fail. So how do we assess sensitivity? Does the signal localize to the biologically correct compartment? That's a really easy one, but it's important. We saw it failed for PD-L1 for one group. And then use a quantitative method to optimize the assay for signal to noise. This is a second really important concept that I think if you're going to validate on a slide, you can't estimate. Human estimation is unreliable and will probably fail, and I'll show you a graphic example of that that's occurring while we're speaking right now. So this is how you measure stuff. How do you measure stuff? Because if you're going to do a quantitative method, it's really expensive to measure 100 slides quantitatively, but it's almost not expensive at all to measure a tissue microarray. So we've introduced the concepts of tissue microarrays as called index arrays. That is, you use it not to measure a population, but as a test series. And so we make all kinds of index arrays in our lab, and you get to design what's in there. So in this case, these are all high PD-L1, these are intermediate PD-L1, and these are low PD-L1 lung cancers. And then here's some cell lines, and here's some placenta control, and some lymph node control, and a range of expression of cell lines. Here's a similar index array for HER2. These are all 3+ fish amplified, 2+ fish amplified, et cetera. So you design the array, and then that becomes your population on which you can test reproducibility. I know of no better way to test reproducibility than using this platform. So sometimes, though, you might say it's hard to test reproducibility if you don't have access to tissues, because I'm a pathologist, I have access to tissues. This is now commercially available. So these are all cell lines, isogenic cell lines, so they won't wander, and they produce defined amounts of PD-L1 under control of non-PD-L1 promoters, under control of exogenous promoters. And so this is, I think we're going to see more and more of this. I just heard of another company that started up and a third company that's now trying to produce these index arrays. And this could be very helpful for us as we think about antibody validation strategies and we make guidelines for strategies is to use these tools. That is, regular tissue microarrays that have actual patient tissue in them, or recombinant or cell line tissue microarrays that have defined amounts of recombinantly produced protein of interest. And can you use one for all different proteins? No, not really. In fact, in our lab, we have an index array for every protein we're going to validate. Can we cross-use them? Yes. We have our lung cancer index microarray, and we use it for multiple markers in lung cancer, but that was good fortune. That is that they worked for more than one, and we had it there, so we tried it. So this is how you do a quantitative titering. And I'm probably going to offend some engineers in the audience because I heard signal to noise actually has to do with variance. And I use the term signal to noise loosely here because what we're trying to do is get the signal of the top 10% of the patients. Those are the highest ones on these index arrays. So whether it's a 15-spot array or a 100-spot array, you're going to have the top 3 or 5%, and that's going to represent your signal, the red line. The blue line represents noise. If you put enough antibody on any piece of tissue, you will get signal, I guarantee you. If you use a high concentration of antibody, some will bind non-specifically to something, and that's the noise. And sometimes some antibodies, especially if they've sat around for a while and there's aggregation going on in the tube, there'll be a lot of noise. And sometimes there's not much noise and you don't have to worry too much about it, but if you plot the noise, and then you do a ratio of the signal to noise, you'll find your optimal antibody concentration for that day, for that usage. And sometimes that's highly reproducible. You can do that same optimization over periods of weeks and months. Other antibodies, and I was talking to Roby about this, are just genetically less stable. That is, the antibody itself has folds and whatnot in its hypervariable region that it's not going to last as long. as an antibody that doesn't have that same exact conformation. So here's an example of optimization of titer of signal to noise. And if you use the antibody here at the peak of signal to noise, this is for PD-L1, and this is actually for an antibody called SP142, which is one of the 4 key antibodies for PD-L1 testing. And if you use it at this concentration, you'll optimize your signal to noise. If you don't, you'll get a result like this. So these are 4 FDA-approved tests for PD-L1 therapies or PD-1 axis therapies. The therapy costs about $140,000 a year. This one actually, there's no specific, the drug hasn't been approved yet. These 3 are all FDA approved. Same piece of tissue, serial sections. This one was not used, I suspect, I don't know, because I'm not inside Ventana, but I suspect they didn't use the optimal signal to noise for this. And as a result, this antibody has about half the number of patients that qualify for this drug than the rest of the antibodies. Here's the antibody used in our lab at the optimal signal to noise. Not the same piece of tissue, but clearly it's not about the antibody, it's about the assay and use of the antibody in the assay. So as we think about guidelines and as we think about sensitivity and specificity, we have to know that we're using the antibody at the right concentration. If you use the antibody at a wrong concentration or a less than optimal concentration, you almost certainly are going to get results that fail validation. So how do we validate? Another way to validate is directly against mass spec. So this is quantitative measurement by mass spec of a bunch of cell lines and then measurement of EGFR. And what you can see is another way to validate is look at concordance. So as we get better, it's hard to see the R values here, 0.75, 0.81, 0.83, 0.88. So as we come to the optimal concentration, our data gets more reproducible. Another way to prove that it's important to validate antibodies, but it also shows that when you use antibodies at the wrong concentrations, you get worse data compared to things like mass spec. And this is those signal to noise ratios. In fact, that antibody, SP142, was the best for signal to noise when used at the right concentration. So that's what I want to say about sensitivity, about specificity. Western blots are a good start. Sometimes that's where we end, but they're neither necessary nor sufficient. Progressive expression of cell lines is a great way to do it, or knockdown or negative cell lines or knockout mice. And another good method is reproducibility, is to confirm with a secondary antibody. So I'll show some examples of that. This is just progressive expression of estrogen receptor, so allowed us to do validation, and I showed you the problem of not doing that in a previous work. This is just an example of knockdown, where if you knock down the protein that you're interested in, the signal should go away, and that gives you antibodies specificity as well. Specificity failure, techniques that I've heard that I would encourage us not to use as part of our validation guidelines. Peptide competition, you can see that suffice it to say, it's not a comprehensive way of proving specificity. Omission of the primary antibody, that's good, but it doesn't prove specificity. Specificity failure is most often due to not the absence of target binding, but binding cross-reactivity. And that's a real challenge. And it may be revealed only when the antibody is used in an application or an assay in which it will ultimately be used. So you might not be able to prove it until you look at a population of hundreds or even thousands of patients. And here's an example. Well, not quite yet, or maybe that one disappeared. So also the next comment is reproducibility, and reproducibility is with a complementary reagent, different days, different lots, different products, and different operators. That's robust reproducibility. So we have to think about what's reasonable to have reproducibility and what costs $10,000 to prove and can't be done on every antibody. And then finally, Andrew will talk about this later. We'll have a whole session on sequencing of the binder interaction domain. So this is an example of a second extracellular domain antibody for HER2, and 2 antibodies are better than 1, and it really validates. And you can see a good regression in real human patients in a number of cancers with 2 different extracellular domain antibodies. It would be hard to figure out a way that either of these antibodies could give this result if they weren't, in fact, specific. Here's an example of reproducibility that we routinely do in our specialized translational services lab, is when we're getting a new antibody from a client, we say, okay, how reproducible is? So we do we plot the coefficient of variation data between different days. And so here's regressions between days, just using a test array. This one's YTMA 279, which has a few, about 30 or 40 lung cancers on it. And you can look at the reproducibility and calculate the coefficient of variation. It gives you the kind of confidence that you can't get by estimating things by eye. And then, well, I lost a slide there I wanted to show. Anyway, so what are we here for? I wanted to not spend too much time. That's all the time I'm going to spend on the concepts, but I think that we need to think about the conceptual pillars. And now I want to talk a little bit about a summary of what the International Working Group did. In some ways, I'll put on my Matthias Uhlen hat, and as he led a group of about 14 or 15 scientists who thought about this hard for a while. That's a pretty small group compared to this group, and it's a really small group compared to the users of antibodies across the world. So did we get it right? I think we got a really good start. I'm quite impressed with the results, but I'm sure there's going to be more to do. Should we have, and so we'll talk about reviewing, expanding, or focusing these conceptual pillars of antibody validation. Determine levels of validation. We talk about this. Do we have different levels? What are the criteria? Do we do some sort of unique identifier? Where is the validation done, and who should do it, and how is the validation monitored or enforced? And so here's this is, I think, in many of the handouts here, and some of the, and hopefully all of you have seen this protein, the 5 conceptual pillars that we came with, and I don't need to spend too much time on that, but just to think about what is a genetic strategy. It's actually any time that you've done genome editing or RNA interference, and this is an example of knockdown, and the criteria is the signal goes away. Again, this has to be, if this was not done at the right antibody concentration, this data would not show this result. Orthogonal strategies, that is a second or independent method, preferably not using an antibody, in this case with mass spec, showing mass spec with the same amounts over a series of different samples. But I think we might want to think about expanding this to other modalities, that is even if they're both antibody dependent, having orthogonal modalities might also be useful. Independent antibodies, and this is something I already showed you, when you have 2 epitope adjacent antibodies and you're fortunate to have that situation, it's a method of high quality validation and highly likely to be successful. And then the expression tag proteins, where you actually have an opportunity to put some other tag. In some ways, this is kind of in the group of dual epitopes or adjacent epitopes, but it's a recombinant approach to a second epitope that's unrelated to the nature of the protein. And then the last was immunocapture followed by mass spec, a more specific mechanism, and can give you information via an orthogonal method, but may not be practical for all applications. So and then with Len's help, we came up with even more conceptual pillars, and this is, I think we'll spend more time on this as we go through, but there's the 5, and then cross-application predictive strategies, spatial localization or fractionation strategies are added to this, so these are things we can think about as other methods of validation that may be useful. And then we'll talk ultimately about who's responsible and what we're going to do here. I would finish my talk by asking you all to think for a minute about what you really want to accomplish here. And as you think of that, I hope that you're not thinking, I'm going to save my company or we're going to get done my agenda, because that's not the goal of the conference. The goal of the conference is really for us to think hard about scientifically what's the most sound method, and then see if we can make all our agendas fit into that most sound method. And if we do anything but that, what we do will not succeed. It won't gain impact. There are standard practice guidelines there are many practice guidelines in the clinic. The NCCN does almost nothing but produce guidelines for how we care for patients. There are other guidelines that are in the scientific and clinical world. The REMARK guidelines, which are guidelines by which biomarker papers are judged before they're published. There's the ASCO CAP guidelines for for estrogen receptor and for HER2. There's the guidelines for testing patients with lung cancer put out by ISLAC and AMP. There's a lot of examples of guidelines in the literature that have worked and have changed fields and improved patient care. And I think we have a shot at doing that, but we need to think about a few things as we do that. Think about what's scientifically best, not what's best for you. businesses that sell antibodies provide an important service, and their needs need to be balanced with the needs of a scientist in the room in the discovery science. Be open and be critical, but be respectful. Thank you.